Okay, wwonderful. Well,
Right.
we have our second speaker of our Palo Alto Parkinson Support Group meeting. It's Bianca Palushaj. Is that how to pronounce your last name?
Palushaj.
Palushaj. Okay, very good. And she just recently completed, was it a neurology residency?
Yep. So I'm in residency now, finishing in June, and I'm going to be staying at Stanford for fellowship in movement disorders.
Fantastic.
So yeah, thanks for having me.
Yeah, and so probably anybody who comes to Stanford will maybe be seeing you in clinic as well.
Exactly. That's
Okay, good.
correct. Yeah.
Get a preview of Dr. Palushaj's. So Dr. Palushaj is going to be talking today about just giving a brief overview of the microbiome as far as Parkinson's disease is concerned. And she's going to be talking about some upcoming research going on at Stanford on this very topic. So we're glad to have her. And if questions come in, please just proceed as with our last speaker. Just type your questions in the Q&A or the chat box and I will ask them. And is it okay to interrupt you?
Yes, absolutely.
Okay, perfect. Okay, great. Wonderful. Take it away.
All right. So as Robin alluded to, I'm going to just talk about the microbiome and Parkinson's disease, what we know and what we don't know. And all of this is in the context of a new study that we have at Stanford that we are very excited about and actively recruiting for. So first, let's just define what the microbiome is. This is a collection of microorganisms that live in us and on us, and it consists of bacteria, viruses, fungi, and archaea. And these numbers here are hotly debated and ever changing, but it's estimated that the number of bacterial cells outnumber human cells by 10 to 1, and that bacterial genes outnumber human genes by about 1,000 to 1. And so traditionally, microbiology focused on studying individual microbial units. And in order to study those, we had to culture them in a petri dish. But in the 2000s, there was an advancement of sequencing technologies that led to this new field of research that we call metagenomics. And so the Human Microbiome Project occurred in 2008 to 2013, and this was a project led by the National Institutes of Health that cost us $115 million and included 242 healthy adults and took samples of the microbiome from 5 different body sites. And the goals of this project were to characterize the microbiome and to begin to analyze the role that the microbiome has in human health and also in disease. And so from the Human Microbiome Project, and of course, a lot of research since then, we've begun to understand what the function of this microbial community is, and specifically the gut microbiome. And so now we know that the gut microbiota produce vitamins that we as humans don't have the necessary genes to make. These microorganisms also break down our food and help us to extract nutrients that are necessary for our survival. The bugs are important for teaching our immune systems how to recognize, what is self and what is foreign. And they also produce helpful anti-inflammatory compounds. And they also engage in what we friendly call bug-on-bug warfare, where beneficial microbes might help to fight off other disease-causing microbes. And so this is one of my favorite fun facts from the animal world, is that baby elephants, when they're born, don't have the necessary gut microbiota in order to digest plant foods. And so what you can see in nature is baby elephants, as they transition from a milk diet to a plant diet, they eat the feces of either their mom or other adult animals in the herd in order to colonize their guts with the microbes that can then help them digest and assimilate the nutrients of the plant foods. And so we know, kind of, like I said, about over the last decade, there's just an ever-growing body of evidence that shows that changes in the microbiome composition, what bugs are there, are associated with every disease under the sun. And so that includes Parkinson's, that includes a variety of neurologic diseases, and that also includes a ton of systemic diseases, metabolic disorder, et cetera. And it's been shown that changes in the gut microbiome or disturbances to the gut microbiome is linked to one's risk for developing a given disease, as well as the disease activity, progression, and what we call the disease phenotype, or kind of the flavor, so to speak, of a disease. So that might be whether, for example, your Parkinson's is more motor predominant or not, tremor predominant or not, how much constipation or GI symptoms one has. And so what do we know specifically about the gut in Parkinson's disease, and why might we even think to study the gut in Parkinson's disease or the microbiome in Parkinson's disease? I'm preaching to the choir with this first point here, and you guys know better than I do that 60 to 80% of Parkinson's patients experience GI symptoms years before motor symptoms start. And they've been able to show that alpha-synuclein, or the so-called PD protein that Dr. Miglis was just referring to previously, can actually be seen in the nervous system of the gut that's called the enteric nervous system in the early stages of disease. In animals, they've also shown that when you inject that so-called PD protein or alpha-synuclein into the intestinal wall of animals, you can follow where that protein travels to with a tracer, and it travels to the brain by the vagus nerve. And then there was a nice study in about 2015 in Denmark where they showed that a vagotomy, where you cut someone's vagus nerve, decreases one's risk for developing Parkinson's disease, suggesting that the vagus nerve is essential for the pathogenesis or the development of Parkinson's disease. And so what do we know about the microbiome in Parkinson's disease? Again, kind of to date, to recently, most of the research has been showing the differences in the composition, saying, hey, in Parkinson's disease, we see these bugs, in healthy controls, we see these bugs. But more recently, there's been a shift of focus from composition to function, and trying to not just understand who's there, but what they are doing and how that impacts our human host physiology. And so we've seen that not only is the composition different, but there's a decreased amount of bugs that create short chain fatty acids. And so what does that mean? So short chain fatty acids are a byproduct of fermentation of carbohydrates from certain bugs in the gut. And they're known to be very helpful to the colonocytes, which are the cells that line the inner lining of the colon, and known generally to be very helpful for colon health and gut health. And they've also been shown recently, they as in short chain fatty acids, have been shown to have an impact on microglia cells, which are the immune cells that live only in the brain and have been implicated in the pathogenesis or the development of PD. In addition, the bugs that live in the gut of Parkinson's patients have also been shown to have an increased production of inflammatory metabolites. And there was one study that showed that there's a protein called curly that's produced by E. coli, the bug that lives in 99% of our guts, and that can induce the aggregation of alpha-synuclein, leading to insoluble tangles that are then toxic to neurons. So again, the summary of this is that we know that the composition is different. What can we learn about the function? And we're already starting to see that the bugs that are either increased in Parkinson's compared to healthy controls or decreased in Parkinson's compared to healthy controls are having an increased inflammatory effect and a decreased production of these beneficial metabolites like short chain fatty acids. I see, Robin, that there's one question. I don't know if you want me.
Yes, it's a little bit of a backtrack. Maybe you could, so the question is, what's the consequence of cutting the vagus nerve? You mentioned that study with the vagotomy. It was in Sweden or something?
It was in Denmark, yeah. It's a good question following Dr. Miglis's talk, because the vagus nerve is the main component of the autonomic nervous system. So you're exactly right. I mean, there's going to be a lot of consequences, and I don't think anyone wants to live with their vagus nerve cut. They're going to have major impairments to their autonomic nervous system, which is your rest and digest. So you can imagine that their digestion takes a big hit. Kind of any of those functions that you think of as the rest and digest nervous system would be impacted.
And I don't recall why the vagus nerve was cut in this particular group of
Yeah,
people.
yeah.
You recall?
It's a good question. I believe it was purely for the research.
Okay, I was thinking it had to do with like they were trying to prevent like ulcers, or they were trying to prevent something, and
In the
that's
peptic ulcer disease, that you may be right. I actually don't, it sounds like you remember better than I do. I don't know for certain, but that would make sense.
that was a very interesting study. But what is the and I'm sorry, I guess I missed it, but what is the relationship then between the vagus nerve and the microbiome?
Yeah. So we think that it is quite literally just what physically connects the enteric nervous system in the gut, which is, a large nervous system that resides in the inner layer of the gut. The physical connection between the gut and the brain is the vagus nerve. And so, for example, in the animal studies, and they've also shown in some human studies, the protein, the abnormal protein, the alpha-synuclein, can be transported along something called microtubules. And so they kind of travel like a highway, the vagus nerve being the highway into the brain.
Okay, very good. And this slide that you're on now is very dense, very a lot of information is on this slide.
Yes.
So it sounds like short chain fatty acids are okay, but the reduced synthesis of them is not okay.
That's correct. So short chain fatty acids are good. And you may have heard of prebiotics.
Yes.
So probiotics are taking a bacteria in a capsule. Prebiotic is taking kind of food for the bacteria that are already in the gut. And so short chain fatty acids are an example of a prebiotic. But short chain fatty acids are good. And it has been shown that in Parkinson's patients, there's a decrease of the amount of bacteria that produce short chain fatty acids. And so therefore there's decreased short chain fatty acids.
Okay, very good. All right, continue on, please.
Okay. Thanks for the questions. Okay, so that brings us, that's kind of the background. So that brings us to the study that we are conducting at Stanford. So we are looking for people with Parkinson's disease and also their, significant other family member that they live with. And we are studying the microbiome, collecting both stool samples and blood samples. I'm not going to read through who's eligible for this, but essentially, healthy controls are not eligible if they have any of these neurologic conditions. And anyone who has inflammatory bowel disease or is currently being treated for cancer, unfortunately, would not be eligible. We're asking for one visit to the research center at Stanford for a blood collection, and then asking for a stool collection. It's a small, you can see my fingers. It's about this much of a stool collection. It's taken from home, put into a preservative, and then mailed back to us with a prepaid shipping label. And then there's also a 20-question survey that's collecting environmental data that will look at any, the impact that diet and animals in the house and other environmental exposures might have on the microbiome. Why would you want to participate? So we're very excited about this because to our knowledge and to date, it's the only study that we know of that's collecting both stool and blood. And so what we're looking at specifically, again, is who's there in the microbiome, and that's what we're getting by sequencing the stool. But we're also interested in what they're doing. And so we're looking at the metabolites, which are all the kind of byproducts of the metabolism of the microorganisms. We're looking at those metabolites in the stool and also in the blood. And then we're also looking at a type of blood cell called peripheral blood mononuclear cell, but this is an immune cell. And we're going to be looking at the gene expression of these immune cells in the peripheral blood. And so again, as you guys very well know, Parkinson's patients have increased inflammation. And so we're trying to better understand kind of how the microbiome and the byproducts that they make have an impact on systemic inflammation. And I wish that we could give more, but we are giving individuals $40 for their participation. And if anyone is interested, I'll leave this up here for contact information. And just briefly, this is our research team. I'm working in the lab of Dr. Ami Bhatt, who is actually in the Department of Hematology Oncology and has a large microbiome lab at Stanford. And who I didn't picture here because the list can go on and on and on, is the movement disorders group from Stanford that you probably well know, Dr. Poston and the rest of her team, who are also helping us with recruitment and such. So this is our team, and here's the contact information, and I'm happy to take more questions.
Okay, great. Is GZMG1, are those people's initials?
Yeah, that's Gabriella Green here. I don't know actually where the GZM, I don't know where the Z and the M came from.
Okay. And okay, and then the 9, is that her cell? The 919
Yes.
number? Okay, all right. I'll add that to our information. And we had one question with a Parkinson's challenge. What can we do nutritionally to support a healthy microbiome and or healthy gut?
Yeah, this is the $1 million question. And, I wish that we had evidence-based medicine is wonderful and we need to practice it, but it's a double-edged sword because oftentimes we stop short at giving recommendations because there's not evidence to support it. And so I think, I don't have much to say that's evidence-based beyond probably what you've heard. There's not a ton of data for there's scant data on specific diets for Parkinson's patients. Overall, as I mentioned, Parkinson's patients have a lot of inflammation, as does any chronic disease. And so a so-called anti-inflammatory diet, of course, is going to be, important. There's even debate as to what that is, but I think that all fields can agree that that's, low sugar, low in processed foods. How can one get short chain fatty acids or prebiotics through their diet? Is by eating fiber, which understandably is hard when you're struggling with constipation and slow GI transit time. But I know a lot of Parkinson's patients will take Metamucil or other fiber supplements, which is good. The conclusion there is that it is healthy for your microbiome to have fiber. So trying to eliminate sugar, eat fiber. Probiotics can't hurt other than what they cost you. I think the consensus by most people in the field is that they're probably not going to help too much and that you might be better off eating prebiotics. And you can take prebiotics, or you can, again, get that through your diet if you just simply Google like what foods are prebiotics. There's a nice study out of Justin Sonnenberg's lab, who's also at Stanford and studies the microbiome, about fermented foods. And they looked at, again, the inflammatory cascade and how immune reaction and basically inflammation is decreased by eating fermented foods. So that would be things like sauerkraut, drinking kombucha, kefir, etc. The one thing you want to be careful about there is that they don't have a lot of added sugar. Let's see, what can we do nutritionally to support? That's mostly it. It's the things that lower alcohol. Does that answer your question?
I'm sure it does. Yeah, it was very complete, I think. And how many people are you hoping to enroll with PD and normal controls?
Yeah. I don't think that we're going to stop enrollment for anytime soon. In fact, we are not going to stop enrollment anytime soon. And what we've actually wanted to do is, right now, this is a one-time, you just come one time, give a sample one time, and then you're done. We would like to, in the future, move into longitudinal, collection. So probably, again, as you mentioned, I'll be in clinic starting July 1st in the movement disorders clinic. And what I imagine and would love to do is just have all of my patients who are willing to pick up a stool sample kit every time they come in and then just mail it back. The microbiome field in general is really lacking on longitudinal data. And we know that the microbiome, changes overall their stability across one's lifetime, but it changes, fluctuates mildly, even from day to day or from morning to night. And so it would be helpful to kind of pick through what's noise and what's signal in this research by getting longitudinal data and tracking individuals over time.
Right. Okay, but people don't have to wait until July 1st to.
No. And so right now we have 80 people who have given samples.
Oh, great.
And we haven't yet started to analyze the data. We've kind of said that at 100, which is relatively arbitrary, then we'll start to analyze data. But no, you can, participate now, and we'll be enrolling for the foreseeable future.
Wonderful. Okay, great. Thank you so much, Dr. Palushaj.
You're welcome. Thanks for having me.
All right. And if you could send me your slides, that would be great.
Yes, I will do that.
Thank you so much. And the slides and the recording of Dr. Palushaj's talk will be posted to our webpage. So thank you very much.